Hello everyone, today we will be talking about X-ray crystallography. X-ray crystallography, as the name suggests, is the study of the crystal structure with the help of X-rays. Now why X-rays are used for studying the crystal structure? X-rays are having a wavelength of around 10 angstroms, which is very good enough to split the light or show a diffraction whenever they enter any crystal lattice. Now from where do we derive the crystal lattice? It can be in the case of organic molecules, in the case of salts, so whatever can be derivatized in the form of crystals. Now in the case of biochemistry, mostly for the biological purposes, two important macromolecules like proteins and nucleic acids, they can be derivatized into the forms of crystals. Now whenever we are talking about a crystal structure means that there is a lattice present. For example, the one which is present in the structure of the common salt like NaCl. So lattice means that many repeating units are present in the structure of a compound, and each repeating unit, so if we are saying that this is the lattice, each repeating unit is referred to as the unit cell. So similarly, in the case of proteins, amino acid build up this lattice, and in the case of nucleic acids, the different nucleotides which are present, the different nucleotides which are present, they build up the nucleic acids. Now, whenever we are having a crystal, so a general representation of the technique is that whenever an X-ray beam, it hits the crystal, maybe for a protein or a nucleic acid, the light gets diffracted. That is, it is reflected in all the directions, and once it gets reflected in all the directions, it falls on this detector and gives certain spots which are useful for determining the structure of the macromolecule later on. Now, how does it happen? Whenever we have X-rays, they will be falling on this crystal. Each crystal is made up of unit cells, as I told you, and each unit cells are having certain important dimensions. For example, three-dimensionally, three sides A, B and C, and secondly, each side is having specific angles. Now, crystals are of different types based on the dimensions of these. For example, in some of them A is equal to B but not equal to C, alpha is equal to beta is equal to gamma is equal to 90, and so on. So this is how we define these crystals on the basis of these dimensions. So they are of different types like cubic crystals, orthorhombic crystals, then tetragonal crystals, monoclinic crystals, and so on. Now all these crystals, they will be having different types of X-ray diffraction patterns, and now why these molecules can be used for X-ray diffraction studies, if we consider that these molecules or crystals, they are arranged like this. So if we represent the different atoms of a crystal or of a unit cell like this, each of this is representing a separate plane. For the X-ray, each represents a different plane, and whenever a light strikes this different plane, I have represented these planes with the help of red dots. So 1, 2 and 3 lines, they are representing the different planes that are made up by the crystal lattice. So whenever a light falls, these are the X-ray beams, say A and B. So whenever the light falls, it is diffracted from one plane. This one is from this plane, say A, small a, and if we consider this as the second plane or B, the other beam, it strikes this B plane and it is again diffracted. Now suppose observer is present here. Observer will not see these two waves as two different waves but as a single wave. Now when he will be observing it as a single wave, there are two types of phenomena which are occurring in this diffraction. One is constructive interference of the waves, another is the destructive interference of the waves. As the name suggests, and as we know that each X-ray, it has a wave nature, it is an electromagnetic radiation. So each crest and each trough, so if we start from here, crest and a trough, it signifies a wavelength. So when it signifies a wavelength, this crest, this trough. So suppose there are two different X-rays, these ones, if we consider them like this, the diffracted rays, if we consider them like this, so if the crest is overlapping the crest and trough is overlapping the trough, if this phenomena is known as superimposition. If we superimpose them, then these are said to be in phase and the amplitude of the resultant waves will be twice as that of the individual waves. On the other hand, there is destructive interference. As we can see, the crest is overlapping the trough and trough is overlapping the crest. So in this case, it is known as destructive interference, and if the amplitude of both these waves is exactly same, what will happen is that we will not observe any wave. Or the resultant wave, it will be having an amplitude which is far lesser than the ones which are contributing to the resultant waves. So now, whenever we are going for the X-ray crystallography, what will we prefer? Constructive or destructive interference? We will be preferring the constructive interference. So whenever we have to carry out the X-ray crystallography for any crystal, we have to fulfill a condition which is known as the condition for the Bragg's law. Bragg's law is nothing but it is the condition for constructive interference, only the condition in which we will see the maximum amplitude for the resultant waves. Otherwise, the waves which we will be observing or the resultant waves which we will be getting, if they have a destructive interference, we won't be able to observe them, or we won't be able to observe them with clarity. Now, if we say A and B, these are the two waves. A comes, it goes to the plane, it gets diffracted, goes to the observer. One thing. B, it goes to the second plane, that is small b. We are representing here. It is again diffracted from the plane. But if you see, B has to cover a certain more distance as compared to A. Now what is more distance? One is this one, another is this one, and then it goes and meets the first wave, that is the A wave. Now whenever this angle theta is the angle which the wave, incident wave, is making with the plane. So theta is the angle which incident so this is also theta, and whenever we make a perpendicular here, if we make the perpendicular here, this distance, this extra distance comes out to be d sine theta. So one is this distance, another is this distance. So this means this particular wave, B wave, has to cover a distance of 2d sine theta. 2d sine theta, and always the wave has to be in constructive interference. When it has to be in constructive interference, it means that the integer multiple of the wavelength should be equal to 2d sine theta. This is the condition for the Bragg's law. This condition needs to be satisfied whenever we have to carry out the constructive interference and in turn the X-ray crystallography of any of the crystals. Alright. So now, first of all, how crystals are obtained. There are many methods by which crystals can be obtained. For obtaining a crystal, the most important and the easiest method is that you obtain the crystal with a supersaturated solution. Supersaturated solution, one of the methods is hanging drop technique, in which what you do is that there is a precipitant and the protein is here. Now, if it is a saturated solution, slowly it will lose its water and the crystal will be formed here and the crystal will be growing here. This is a hanging drop technique. It can also be formed in the capillaries. It can also be formed in the oils, micro drop technique. So, with the help of these techniques, first of all, you should be having a crystal. Now, what should be the properties of this crystal? First of all, it should be around 95% pure. Now, how do you know about the purity of the crystal? Once the protein or the nucleic acid has been derived, some of it would be used for carrying out the procedure of electrophoresis, in which the purity can be determined. It should be very concentrated solution and at least 500 microliter of it should be there. because at least some crystals should be there which can be placed in the apparatus and the X-ray crystallography can be done. Now, once we have obtained the crystal, after obtaining the crystal, now the crystal is placed on a device which is known as a goniometer. Now, what is a goniometer? If you have seen in certain movies, the goniometer is a kind of device. In the movies, you must have seen that the diamonds are placed on a device like this. Diamonds are placed and it keeps on revolving. So, it is around 180-degree movement, and goniometer is the device which can move it like a spindle in all the directions. Why it needs to move in all the directions? Because X-rays have to fall on it. X-rays have to fall on it. Now, once the crystal is in line with the X-rays fall on it, then there is a LED screen. Why LED screen is there? Because very high intensity of light, if it goes to the detector, it will lead to a very, you can say, overexposure of the detector, and to avoid this, the intensity is controlled with the help of a LED screen, and then in the conical form, it will form a pattern. So this is the kind of pattern which we get. We will be getting a pattern of repetitive spots. Here I have not shown, there can be certain specific patterns which are repetitive. Then with the help of a technique of Fourier transform, which is a mathematical algorithmic technique, it is transformed into a map which is known as electron density map. Where the electrons are more, it will form a kind of map. Now indexing needs to be done. What is indexing is that now we have got an electron density map and we know the real structure of the macromolecules. It is made up of carbon, hydrogen, oxygen, nitrogen. Then we start fitting in the, physically we start fitting in the structure of those, what you say, overlapping the structure of carbon, hydrogen, what kinds of bonds are possible, and then we detect the actual structure of the compound. So this is all about the X-ray crystallography. Just to revise once more, X-ray beam falling on the crystal giving rise to a diffraction pattern. Now diffraction is a bit different from the phenomena of reflection. In the case of reflection, what happens is that the beam goes and is reflected from any surface, whereas in this case there is a certain slit through which it goes. So there are certain distances, as you can see, small distances between the intramolecular surfaces which serve as small slits through which they pass and give rise to the diffraction pattern. Then a condition which is known as Bragg's law, it needs to be satisfied, and then once it is satisfied, the constructive interference takes place. The crystal is loaded onto the device which is known as goniometer. Through the LED screen, it passes, the light passes, and the reflected or the diffracted beams, they fall on the detector. With the help of Fourier transform, we get the electron density maps. Once we have the electron density maps, we know the structure of the different carbon-hydrogen bonds, carbon-nitrogen bonds, carbon-oxygen bonds. Then we try to fit in the dimensions of those bonds into these electron density maps, and finally we built up the structure of any unknown protein, which is very useful for the nucleic acid or protein determination of the structure of any new protein isolated, determination of structure of any new viruses which are isolated, or bacteria, etc. Thank you so much. Please let me know if you like the video.